Thank you, Professor Silvio. I'm going to now introduce the first session. It's actually my pleasure to welcome you all to this first session of the first Human Cell Atlas Latin American meeting. This first session will bring together essential topics that define the HCA initiative, and we have some amazing speakers. We are in fact very lucky to have today both Aviv Regev and Sarah Teichmann, who are co-founders and co-leaders of the Human Cell Atlas initiative. Sarah co-founded in 2012 the Sanger EBI Single Cell Genomics Center that pioneered single cell genomics, and she's currently the head of the Cellular Genetics program at the Wellcome Sanger Institute. She has steered the program towards a strong focus on cell atlas technologies and with the aim of understanding human health, development, and disease. Aviv is also a pioneer of single cell genomics, and her ultimate research goal is to combine experimental and computational approaches to decipher how molecular circuits function in cells and between cells in tissues. She is currently the Executive Vice President and Head of Research and Early Development at Genentech. I welcome you both, Aviv and Sarah.
Thank you. So welcome everyone, and we're really thrilled to be here, even if it's all virtual these days. At least it gives us the opportunity to all be together. I'm going to open this talk that Sarah and I will present jointly. The tradition in HCA meetings is that we always split the talk. Sometimes I open and sometimes Sarah does. So it's my pleasure to be the opener this time. I'm going to start with both Sarah and my disclosures. We have involvement in multiple companies, and in my specific case, I'm now an employee of Genentech and Roche. So as you've already heard, the basic unit of life is the cell, and each of us is made of about 37 trillion cells, which come in many different types, like neurons and immune cells and muscle cells and fat cells. And of course, at first glance, one might think that every cell in the body is the same, because after all, there's the same DNA and the same set of instructions, except that, of course, we know that the cells are quite different from each other. So unlike in the case of the human genome, where for one person you can assume one genome, for one person there are many different kinds of cells. And the fact that the cells have the same DNA instructions but use different parts of them really has critical implications when we start thinking about the genetic causes of disease. Because if there is a genetic variant that confers risk for disease, all of the cells in the body are going to carry this variant. But of course, the disease will only manifest in those cell types that express this gene. And so knowing our cells is going to be essential for understanding how the genes that cause disease act. The only problem is, of course, that we don't really know our cells. If we open any resource like Wikipedia or a textbook, it might highlight a few hundred cell types overall. And these would be indeed things like neurons and some subsets of neurons, immune cells and different subsets of immune cells, and so on. But of course, if we look just at one part of the nervous system like the retina, there could be hundreds of subsets of neurons in the retina, and of course, many thousands maybe in the brain, and then who knows how many in the rest of the body. And that is one of the big open questions for us, because what we really need is some map or an atlas that would have clear coordinates that would let us know all of our cells and their characteristics and what they do. And so one might ask, what might be the coordinates of the map? Historically, biologists have defined cells using many different means. They might look at their structure, their function, their molecules, one kind of molecule, other kinds of molecules. And so one way of unifying this information is by searching for a joint set of coordinates. And the level of expression of each gene in the genome can serve as these unifying coordinates. And so in the context of a map like this, if we imagine that there is only 3 different genes, each would be captured by one axis, and each cell would be a point in this 3-dimensional space. Of course, we have 20,000 or more genes, and so each cell is actually a point in a very high-dimensional space, and computational algorithms help us navigate this. And so then that only leaves the question of where would the data come to position cells in a map like this. And the honest truth is that for a long time, there was a technological barrier to actually build maps at this level of resolution. until the recent advances, both in single cell and more recently in spatial genomics. And so until recently, when we wanted to profile cells, we had to make one of two choices. Either we could look at very few variables of genes and we would do it in a tissue context, or we would look at all genes, but we would do it using bulk genomics, which we like to analogize to a fruit smoothie, if each cell is one piece of fruit. With single cell genomics, we are able to measure the characteristics of each cell individually using profiling techniques, such as RNA-seq or ATAC-seq. And in spatial genomics, we get to do that even in the spatial context of the tissue. And so the first set of advances that occurred in the field was indeed in the area of single cell genomics. And one of the transformative steps that has happened in that area is the ability to go to a massively parallel scale, which we can do using different kinds of technological means. For example, one can use droplets, but with more recent advances, for example, from the work of Alex Shalek, one can also do it in the context of lower tech devices that can be deployed even in the field. And the combination of the resolution of the technology and the scale of the technology really allows us now to build atlases of tissues of high complexity and with very high resolution. In the context of disease, this opens up a very large set of questions that we can answer, including the cells in which disease risk genes act, which cells are disrupted in their physiology in the disease, how are their gene programs changed, what are their functions and their modules, how are the communications between cells in healthy tissue disrupted in disease, and even what might be the effect of drugs. But in order to understand all of these things about disease, we first need to know what health looks like. And so if having a map would be so useful, the reasoning was, let's make an atlas of all human cells. And that became the mission of the Human Cell Atlas, to create a comprehensive reference map of the types and properties of all human cells, which are the fundamental unit of life, and use it as a basis for understanding, diagnosing, monitoring, and treating in health and disease. And we sometimes fondly allude to it as a periodic table of our cells, and we try in that to convey two concepts. The first is that we would like to have a comprehensive view of all of the different kinds of cells. And the second is that it might be possible in a data-driven way, in the same way that Mendeleev had a periodic table of the element, where he could even predict elements that were not yet experimentally observed and understood the underlying principles of elements, that we would reach a similar understanding of the cells in our body. As a path along that, we need to look at things at many different levels of resolution. And so we often allude to it also as a Google map of the human body, but instead of an Earth view and, you know, the overall roads, and then the high resolution of a street view, and overlaying it with different things like where are the gas station and what are the driving patterns. In the case of single cell genomics, this is provided by the in the case of the Human Cell Atlas, this is provided by the combination of single cell genomics technologies and spatial transcriptomics and spatial proteomics technologies that give us a view of different molecular layers as they are in individual cells and as the cells are organized in space, both at the resolution of a local microenvironment, histology, microanatomy, and macroanatomy. And so with this, I will turn to Sarah, because humans are diverse and so are their cells. And the Human Cell Atlas is not an initiative of one lab or one institute or one country. It is an international initiative of the entire scientific community. And Sarah will start to describe our initiative next.
Thank you, Aviv. As Aviv mentioned, we view the Human Cell Atlas as a global effort. It's much too big of a project for any one person, one institute, one country to do on their own. And it's a tremendous pleasure and gratifying to us that the HCA from the beginning has been a grassroots initiative growing up through the scientists who are involved. And so we welcome you to join HCA by registering at humancellatlas.org/joinhca. And as you can see, we're now at 2,000 individual members from 74 countries. And in Latin America, we have 65 members from 45 institutes. So let's increase that number by having a lot of you join, and you can join as an individual member or with a project. Next slide, please. So going back in terms of the history, we are now in September 2020 at the Latin America meeting that you can see on the far right. And the origins of our meeting series go all the way back to October 2016, when we had our first kickoff meeting in London that Aviv and I organized together, and that had about 100 members, so similar in size to this meeting. What it would be great to make you aware of is that we also have now, especially during this challenging period, we have a lot of online meetings and seminars like this one. And we invite you to join the development seminar series, for instance. We also have a COVID-19 symposium, and you can find out more about the meetings on the humancellatlas.org website. What you can also see here is that we have meetings dedicated to specific topics like development, also to specific technologies, including jamborees and hackathons and that sort of thing. And the other thing to emphasize from this timeline is that in the history of the project, we are now really in an active data collection phase, and I'll come on to describe that later. Next slide, please. At the same time, it's important not only what we do, but how we do it. And so in our white paper that you can read on the archive, we laid out principles of how we wanted the project to go about the science. And that includes quality, of course, flexibility, the community, equity, diversity, and inclusion, both in terms of the samples and subjects, but also in terms of the scientists, transparency and open sharing of data, code, and also protocols, respecting transparency and openness while respecting privacy and ethics and local and national regulation, with a constant eye and dedication to technological and computational innovation. Next slide, please. So going on to the analysis, the individual working groups and networks in terms of the structure of the community, it's a pleasure to introduce the analysis working group, and that's co-chaired by Donna Pear and John Marioni, and has 8 other members. And what they organize is data analysis teams, meetings, and jamborees. And basically, you're welcome to engage by going to the website and seeing the emails. And what they engage with is experimental designs, analysis frameworks, and also advising the data collection to the data from data collection to the data coordination platform on computational analysis questions. Next slide, please. There'll be a talk later in this meeting on the data coordination platform. And of course, that's a really important part of the project, because that's where the data is ingested and then served, so and organized in a manner that can be queried. At the moment, the data coordination platform contains all open access data, so data that can be shared completely freely from the raw level. And going forwards, it will include support for managed and controlled access data, which a lot of the human data is under that sort of ethics. It's also worth mentioning that, of course, one of the major intellectual challenges for the Human Cell Atlas is cell annotation, and there's a computational support platform that's under development to support that, led by Peter Karchenko with Aviv, Fabian Theis, and John Marioni. Next slide, please. The Standards and Technology Working Group is another working group that we've had from early on in the project, and it's chaired by Holger Hein, Ulrike Rosenblatt-Rosen, who will speak later in this meeting, and Xiaowei Shuang, and it brings together groups who are interested in comparing methods, disseminating existing methods, and sharing the newest methods. And so again, the contacts, they meet monthly, and the contacts are online on humancellatlas.org. Next slide, please. In this context, Musa Mlanga, Partha Majumdar, who can't be with us today, and Alex Shalek have already been mentioned in the introduction by Lucio and Silvio. And we're really delighted and grateful for their leadership in terms of increasing and extending the equity and diversity of the scientists and the samples in the project. Next slide, please. The past half a year or so has been very challenging at many levels, and that includes the Black Lives Matter movement. And I'm really proud of the HCA community and how it came together and disseminated the statement and had discussions around this. And these continue in the community to now, to the present day. Next slide, please. And one of the commitments that came out of this was to expand outreach programs in underrepresented countries in the HCA community around the world, to implement a diversity audit, both in terms of the membership, the leadership, scientists, and samples, so that we know where we stand and we can do things to address the gaps. And then the third activity in this area that we've committed to is working with the funders to encourage the earmarking of funds to increase diversity of scientists and samples. And one of those examples is the Trans-Zuckerberg Initiative's commitment to increasing the diversity of samples that are studied in all of their human cell atlas seed networks. And those grants actually close next week. Next slide, please. So the way we planned those actions was by having a survey, which you can see online, and some of the actions that are planned and that have come through the context of that survey are travel stipends to increase participation from people around the world, and also commitment to organizing meetings to reduce travel expenses. And one example of that is the Vietnam workshop next year, which is dedicated to training on experimental technologies and computational methods. Next slide, please. It's also maybe useful to know about the ethics working group, and the goal of that group is to develop a toolkit and to develop support for ethics around the use of human tissues. This isn't specific to individual countries, it's a core consent toolkit, and that's been developed by Bartha Knoppers at McGill, together with her team, including Emily Kirby, and the co-chairs Barbara Walden, Rodrigo, and all the members of that very active working group. And so those toolkits are available online to support with the ethics submissions and so on. Next slide. On the HCA doesn't only have these technology working groups, but also biological networks for biomedical scientists who are dedicated to specific tissues or specific organs of interest. And we have we have 14 of these that you can see here, and they each have coordinators and dedicated emails, and we invite people to join these groups. And they include different adult homeostatic tissues, but also you can see human development is the third from the left in the top row. And you can also see organoids at the bottom. So one of the major translational impacts of the human cells will be the implications for tissue engineering. The organoid working group is dedicated to that topic. Next slide, please. So what I'm going to go on to now is to give you a snapshot of where we stand in terms of single cell RNA sequencing data for suspension cells from all these different tissues that you've seen in the previous slides, and to give you an impression of where we stand in terms of our science before going on to COVID-19 research. Okay, next slide, please. So you can see the different organs where we've made really exciting, significant progress. Here you can see for many of the organs that we're showing here, so liver, kidney, skin, airways, gastrointestinal tract, human development, there are samples from many hundreds of individual human donors, and there are often a million or over a million single cells that have been profiled. And what that means is that we're now getting a real grip on the diversity of cell types in all these different tissues. And so we're really starting to get towards that periodic table of cells that Aviv mentioned early on in the talk. Next slide, please. Now, COVID-19 has gripped the world over the past 9 months or so, and the human cellist community has really picked up the glove and risen to this challenge. And you can see here that the community has profiled hundreds of thousands, if not millions, of cells from different tissues from infected patients. Next slide, please. Here there's more data on other tissues, breast, heart, brain, female reproductive tissues, pancreas, and blood and immunity on the left. And if you, next slide, then you can see that for COVID-19, of course, the blood is of particular significance, and there's almost 4 million cells from COVID-19 infected PBMCs that have been profiled to date, and that's starting to make real progress on understanding the antiviral immune response to different patient cohorts, for different patient cohorts to this disease. Next slide, please. Coming back to the Healthy Reference Human Cell Atlas, I just want to give you a few highlights from the lung and the airways, and this is data on the reconstruction of the cell lineages by Pascal Barbry, showing the progenitors of the secretory and ciliated cells and the intermediate uterosomal cell states that can transition between them. There's also been data from Herbert Schiller's lab with Fabian Theis on on lung aging in the mouse, and also changes in fibrosis. And then work that from my group in collaboration with Martin Nawain and Herbert and Fabian on novel T cell states, and also epithelial transitions in the bronchi in the healthy lung and in asthmatic patients. Next slide, please. The lung is of course an area of lots of research and lots of interest because of its implications for disease. And so there's also really interesting work on fibrosis from the Kaminsky lab, and you can see the bioarchive links here. And you may have seen the Atlas of the Lung that came out in Nature earlier this year. And then the human branching tree from the upper generations down to the parenchyma and the alveoli, in progress with spatial analysis from J. Regev and Aviv here on the right-hand side. So there are many different, the take-home message is that this massively complex organ is being tackled by many different groups from different perspectives, both in health and disease. And that's really important at this particular point in time with the COVID-19 pandemic in the airways. If you go to the next slide. And so what you can see is that we're getting from a draft atlas where we have sort of hundreds of millions of cells, and that's where we are at the moment, and where we aim to get is a really comprehensive atlas up to billions of cells and with deep spatial information over the next 5 or 10 years. Next slide. And so what I hope is this example of the lung has also illustrated is that the Healthy Human Cell Atlas is a really great framework and partner for, if you go on to the next one, for human diseases. And I'll illustrate that now in the next few slides on the example of COVID-19. If you go on to the next slide. So just a quick shout out to the funders too. So also the Human Cell has been funded by many, many different funding bodies that you saw flick through very quickly on the previous slide. And they're really, we're grateful to the partnership with all the funders from around the world who've been supporting us and making this really vibrant community possible. So early in the year, when it became clear that we're faced with this, the pandemic of the SARS-CoV-2 infection across the world, the Human Cell Atlas community rose to this challenge and together profiled the expression of the viral entry protein. So the ACE2 enzyme, that's the receptor for the virus, and then the accessory proteases. And what this revealed was that there's the known alveolar type 2 cells that were published as early as 2004 as the coronavirus entry point in the parenchyma and the alveoli of the lung. But there are also many other cell types that the Human Cell Atlas community has described that were not appreciated as viral entry points. And those include many cell types that are in barrier tissues that are known to be really important for the early stages of infection and for transmission. Next slide, please. And so in those early days, the Human Cell Atlas community realized that we need to provide the library of all those cell types to the community as quickly as possible. And everybody pulled together from around the world and shared sort of unpublished data, also very generously from many different tissues to make that possible. Next slide, please. And so that's already been realized in 3 distinct publications. So one on early infection and transmissibility in Nature Medicine, one on model organisms, focusing very much on model organisms, non-human primates, mice, and the interferon response in Cell, and then a third one looking across data, many data sets from humans in terms of the pathogenesis and covariates of the disease. And that's coming out in Nature Medicine soon and is online on the bioRxiv. Next slide, please. And so what those 3 publications have taught us collectively already at this point is key cell types that can welcome the virus because they have the entry points and that are likely involved in early infection and transmission. Also potential routes of and mechanisms of vertical transmission, pathogenesis, in other words, the dynamics of the infection, mechanisms with respect to ACE2 and innate immune responses, the epidemiology with respect to age and smoking, gender, and so on, and then also how model organisms can help us learn about the disease. And that's really all based on the Healthy Reference Atlas. I'm going to hand over to Aviv to talk about the community's study of patient samples, of COVID-19 patient samples now, and finish off. Please, Aviv, over to you.
Great, thank you so much, Sarah. So I'll wrap up by telling you a little bit about what is going on in the Atlas community and in single-cell genomics in the context of individuals infected with and who passed away from COVID-19. And by and large, one can separate these studies to 3 major areas. And you've already seen a little bit about the statistics of the cells that have been profiled from COVID-19 patients. There's a major effort to look at blood samples, at peripheral blood mononuclear cells, comparing patients with mild, moderate, and severe disease, patients who have already convalesced from COVID-19, and using the data both to understand immune cell states as well as to understand neutralizing antibodies and T-cell receptors that are involved in the response. A second major effort looks at tissue samples that are obtained from COVID-19 patients. And these include aspirates, nasal swabs, and bronchoalveolar lavages that you can obtain from individuals, that you can obtain from live patients, and again, analyzing them by single cell and RNA-seq and similar technologies. And then the third effort tries to understand the way that the virus really ravages the body of individuals and can lead to death. And for this, we rely on autopsy organs from COVID-19 patients, and those are analyzed. Here you can obtain, sadly, you can obtain tissue samples from across the body, not only from the interface tissues, so lungs, hearts, livers, kidney, brain, and so on. And these tissues are analyzed by single cell or single nucleus RNA-seq, which is particularly suitable for tissues that are collected at autopsy, as well as, very importantly, by spatial analysis. And I'll show you just a small glimpse from some of these efforts. There's efforts all around the world. This was a tremendous tremendous feat to pull off, and the number of autopsies that can be analyzed in any given location is naturally limited. And so Europe, in the United Kingdom, in Israel, and in other countries. And the 2 that I'm highlighting here are one effort that comes from 3 Boston area hospitals, MGH, Brigham and Women's, and Beth Israel, and the second effort that comes from London, from Imperial College London, from the UK. And these efforts coordinated with each other, shared a lot of protocols, a lot of pieces of advice went back and forth in order to ensure that each can do the best that they can by patients and our understanding of the disease. In each, there's 10 to 20 autopsy samples, a large number of tissues from each of the individuals, and overall hundreds of tissue specimens that can be analyzed. And I'll show you a couple of highlights and images from the Boston area effort. So first of all, to highlight that Pulling this off in the middle of the pandemic is, of course, a very difficult challenge. It requires a great amount of coordination between individual labs on how tissues are collected at the autopsy suite, optimizing the sample preservation protocols, handing them off, and training scientists who have done only single-cell genomics in a traditional lab setting to do these kinds of experiments in the context of a BL3 facility. And you see 3 of our young scientists actually all suited up as they're doing their work. The one in the middle is an expert in BL3 work, but has never done single-cell genomics. And the 2 flanking her have done a lot of single-cell genomics and had to train into a BL3 facility, literally in the middle of the pandemic. And I should point out the HCA has a lot of partnerships, and we're really grateful for some of these partners for having been having rapidly pivoted to allow a BL3 facility to have the right equipment and reagents in order to run these experiments. And so what can we learn from this kind of data? I'll show you only 2 very brief glimpses. From looking at autopsy lungs, we can use the Healthy Reference Atlas both as a source for annotating the COVID-19 samples that we have, as well as for looking in comparison to other disease conditions. For example, this is from pulmonary fibrosis data from the Naftali Kaminsky group that is focused on lung conditions in the context of disease. And this allows us to understand both the cell state differences that exist in COVID-19, as well as the dramatic remodeling of the tissue. You can see the great contraction in the epithelial compartment and the great expansion, for example, of the endothelial compartment in COVID-19. And so there's a dramatic change that happens in the tissue. These analyses are now ongoing across many other tissue types. Here you see liver, kidney, and heart data, all from multiple COVID-19 patients, each of them analyzed and annotated with respect to a healthy reference atlas that was collected as part of HCA. And without the healthy reference, we actually wouldn't have been able to understand what is going on in the disease. As I noted, in parallel, analyses are ongoing to also analyze at the spatial level. One of the techniques that's been deployed is the GeoMX technique made by NanoString, which allows us not to perform individual single-cell analysis, but instead to identify small regions of interest within a tissue. Within those regions of interest, dissect between the regions that are stained by specific markers, for example, for epithelial cells or for immune cells like CD45, and capture the RNA only from an area of interest within a region of interest and profile that RNA separately. So this allows us to look at a particular region, say infected by the virus, which we identify by hybridization to viral RNA, and then from that region distinguish infected epithelial cells from immune cells or stroma within this environment. And so, for example, this allows us to perform a differential expression analysis between COVID-19 high and COVID-19 low regions, and within those regions compare the epithelial cells in them and look at the responses, which in this case are immune-like responses that exist in the cells in infected regions compared to the same kind of cell in a non-infected region. This work is all still ongoing, but it already highlights some responses such as interferon alpha and gamma responses and antiviral responses in the infected cells. All of this data is gradually appearing online, released well before publication, often before submission even of a paper. And here I want to give a shout out to all the portal developers, and in particular to a new release that is coming up on COVID-19cellatlas.org, where one can look at the expression data that is available from these studies. Due to our commitment to, we always have a commitment to rapid release of data, but this commitment is much reinforced in COVID-19 times. And so I'll conclude by saying that we're on a journey from a very fuzzy and early draft, but every piece of information matters, to the first draft that will consist of about 100 million cells, and we're well on our way there, as you could see from the fantastic advances across all of the organ systems, and towards eventually a comprehensive atlas that will consist of up to 10 billion cells at both single cell and spatial resolution across many different genomic molecular modalities. We hope that this would allow us to gradually, color ourselves and understand that we're not just made of a lot of different cells, but understand the fine characteristics of these cells and build a true Google map of the human body. Thank you very much.
Thank you so much, Sarah and Aviv, for this great presentation, for sharing the history of the HCA, and also for showing us how the platform, the collaborators are so ready to contribute with up-to-date things at real time. We have quite a few questions which are very technical, so I think they're going to be answered throughout the meeting. And we don't have so much time for questions anymore, unfortunately. But I have one question here that has been highlighted, and maybe you can try to answer that. The Human Genome Project had a very defined goal, which was to sequence the entire human genome. The project was finished once they sequenced about 2.3 billion base pairs with coverage, etc. But at what point do you think the HCA project will be considered completed?
I think that's a fantastic question, and I will be very open and honest that when we embarked, it was very difficult to answer it, because unlike with the human genome, where we had a sense of its finite size when we started, with cells, we definitely don't need to profile 37.2 trillion, but because we didn't know the statistical characteristics of the diversity of cells with tissues, all we could do is make a back of the envelope calculations at best, and that is where these numbers, the initial numbers, came from. Let me start by alluding to the fact that the Human Genome Project finished in the sense of generating a map of the non-variable part of the human genome. That was only the beginning of the human genetics part of the Human Genome Project, which is still ongoing, because it allowed us to get the invariant part. And as we dug deeper and deeper into human genetic variation, we still conduct these studies. We're still not done with that. And that's the beauty of reference maps, that even when they're incomplete, they are highly useful, and the Human Genome Project is a great example of that. Today, given what we understand about the extent of variation in cells between individuals and the fact that they're actually reasonably invariant, and we can combine information from a relatively modest number of individuals and basically get similar answers. And as we have gotten better at enriching for rare cells, so we don't have to profile the common cells over and over and over again in order to get a handful of rare cells, I think our initial notion of the 100 million cells for a first draft was a pretty solid one. Maybe it's even more than we need in order to get to a first draft. But the truth is that the technologies are so great that we'll probably get it. That we will probably get it no matter what. At the same time, there is a very substantial effort right now to develop a power analysis to understand the spatial diversity of our tissues in order to know how much do we need to sample, not the organ as a whole, but the different parts of the organ, and how much sectional information do we need. And that really varies organ to organ, with the brain probably being in its own special pedestal in terms of spatial diversity, whereas in other organs where there is more repeatability, we would be able to do less.
Thank you. Thank you so much, Aviv, for this great answer. Sarah, do you want to add anything to that?
I was just thinking, I mean, I think Aviv has really covered it, you know, the work with the community is getting a good grip on the statistics of the depth that we need to comprehensively grasp all cell types. What would be beautiful and amazing would be if one day we could take a single donor and basically take sections comprehensively from the head to the toe and do a spatial transcriptomic single cell resolution profiling of an entire human being. You know, that would be the human cell atlas of one man or one woman or both. You know, that is far from being technically feasible and affordable at this point in time. And so I think the estimate that I mentioned during the talk of a 5 to 10 year project, you know, Aviv has mentioned 100 billion cells with the anatomical sampling of the organs, that's really right now what we're looking at to get a human cell atlas. And with that, I'll end and just say thank you very much, Patricia, for your fantastic sharing, and thank you to my partner, Aviv.
Thank you so
Patricia,
much,
and thank you, everyone.
Unfortunately, we have to move on. We have a very tight program.